Good morning, good afternoon, good evening, everyone. This is the fifth session of Talking with Titans, jointly organized by Aquaculture Nutritionist Network and Jeffo Nutrition Inc. I think you all probably know, but Aquaculture Nutritionist Network is a group of aquaculture nutritionists started in 2011, and today we have about 2,900 members worldwide. And today with us, as always, Dr. Albert Tacon, the global icon to a lot of people. And our guest today is Dr. Jesse Trushenski. And we also have to help us in the back, Pumjai from Bangkok. So you have like almost 3 continents, North America, Asia, and also North America, Hawaii, but it's part of Asia somewhere, right? Pacific. Just introduce Dr. Trushenski. I think she started her career in as a professor, as a faculty member at Southern Illinois University, and then moved to I think a little bit of public work, public research, and then now in private with the private industry. I think aquaculture farm, right, or production company, Reverence and Evaqua Farms, as director of science, I guess, right? And she was also, I don't know if you were still president of American Fisheries Society, right?
I just finished up not too long ago. I'm a past president now, which is the best job.
That's the best job. So, yep, we will let Dr. Trushenski say something about, her career growing up and how she became interested in aquaculture and then nutrition, and then I think research focus from the beginning to now, like a farming
Sure.
side. Go ahead, please.
Yeah, thank you, Kabir. Thank you, everyone. It really is an honor to be a part of this series. I'm following in some pretty big footsteps, so hopefully I won't let the side down. But yeah, I think I came to aquaculture as a result of what I call my quarter life crisis. So I was in college and I was studying marine biology. Like a lot of people of my generation, I wanted to be a marine biologist when I grew up. And it just kind of struck me one day that, really if I continued down the path I was on, maybe the best I could hope for someday would be, maybe I could be on like a Discovery Channel special, a documentary someday. And maybe my parents would watch it or my friends and family and they'd say, huh, that's pretty cool. And then they would change the channel and be on to something else. And I realized that while that public education and those documentaries and science communication, that's all really, really important. But it just felt like I couldn't wrap my arms around it. Something was missing, and so I was kind of, I don't know, looking for what my real career should be. And I happened to be taking a class all about algae, and at the end of that class, they talked about kelp farming, and it was like the light bulb went off. It's like, wait a minute, I can use biology, I can work with aquatic species, I could use chemistry and math, maybe a little bit of physics sometimes, and I could feed people. That was something I could really wrap my arms around and feel like this could be a fulfilling career. And so I spent the rest of my time in undergrad learning more about aquaculture. I think every time I had a chance to do a term paper about something, if the topic was my choice, I was writing about aquaculture and educating myself. And then ultimately went to a graduate program that could help me sharpen my skills. And that's ultimately how I came to nutrition within aquaculture, because I did have a specific interest in that area. But really, the way I came to aquaculture was all about trying to find a way to help people and really feel like I could spend my whole career doing something that would matter. So that's how I got here. Kabir, you're on mute, I think.
Sorry, I was trying to. I was thinking that your, introduction would be a little bit longer. But
oh, sorry. Yeah, sorry. I didn't know how much you wanted me to say.
Oh, it's okay. It's okay. Albert, anything to say? Like anything to add or anything you want to say? Like a situation in Hawaii, and how are you doing alone in Hawaii now?
Can you hear me?
Yes.
Yeah.
Okay, no, just that this is the fifth time we've been on this webinar series. It's an honor to be here, and I'm humbled with a guest like we have now with Jesse. And I think it's really nice to have a forum which is open and where people can exchange ideas at all levels. But at the end of the day, we are here to serve the aquaculture industry, the aquaculture feed sector, and so it's important that we continue to do this. So again, I'm honored to be here, and thanks to Kabir, to Jesse, and to Jeff for inviting me.
No, you were the organizer as well.
Yeah.
He's partly responsible for all this.
Yes, yes,
Good or
yes,
bad.
obviously, yeah. He's the big, the switch. You have to have someone is to turn the switch, but he's the switch. So, I will just before we start the formal presentation, I will ask Poomjai to talk about some housekeeping stuff, right, for our audience today.
Okay, so for the webinar today, we will have a record for the webinar, which you guys can get a link later on. But if anyone would like to download the handout, you can go on to the right-hand side to the handout session, and then you can download the handout for this session. And if you have any questions, you can also write your question in the question box, and then we will answer the questions at the end of the webinar. So I will give back to Jesse.
Okay, but I'll just thank you, Poomjai. Today's topic is fatty acid for sure, right? And I think we put a lot of focus on amino acids and protein for sure, because protein is the most costly, portion of our aqua diet. But a lot less focus on fatty acids. And I think one of JC's paper, I think it's 6 or 7 years ago, it intrigued me on these. We talk about the saturated fatty acids, right, and then gene expression stuff. So that was in my head for a long time, and today's the opportunity to learn about it a little bit in detail and in depth. Thank you, Jesse.
Great. Okay. So I think, see if I'm ready to share my screen here. And
you have to go to,
I'm looking for the option to change to a different screen. Sorry guys, it was right there.
No, it's okay. It's okay. It's on top. You have used to a screen, sir.
Yeah, I just have change presenter. I'm going to stop showing and restart.
Okay.
Okay. Okay, Kabir, can you see the correct screen?
No, you have to. No, you have to go use to a screens mode on top of your screen. Can you click that? It will.
Let's try that. Did that do it? Good.
Yes.
Okay, I had to find it a different way. It wasn't the same way this time, but whatever, we're here. We're doing it now. So, okay, well, again, thank you all very much. Sorry for the technical difficulties there. You would think that we hadn't done a practice session. I promise that we had. But yes, today we're going to be talking about fatty acids, a subject that's near and dear to my heart. And as Kabir mentioned, it's something that doesn't always receive a huge amount of attention in fish nutrition, but is like all nutrients, fatty acids are just as important as any other part of the diet. Any nutrient can become limiting, of course, in the formulation. So let's carry on. And again, I can just begin by setting the stage with just a few kind of key facts that have really driven not only my research in fish nutrition, but I think probably everyone's research. And that is that the aquaculture industry continues to rely on marine resources, fish meal and fish oil, as key components of these industrially compounded commercial feeds. Fish meal and fish oil have, of course, always been used in aquaculture feeds, and it's my view that they always will be to some extent. And I think that's okay. Using fish meal and fish oil in aquaculture feeds is a much more productive use of those resources than feeding them to poultry or to terrestrial livestock, or certainly turning them into fertilizer. But the aquaculture industry is, of course, growing, as is the aquafeed industry, and that's increasing demand for all feed inputs, and that includes marine resources, of course. So the bottom line for us is that it's okay to keep using fish meal and fish oil, but to produce the volume of aquaculture feeds that are needed now and in the future, those inclusion rates are going to have to come down even more so than they have to date in order to stretch those limited supplies to satisfy growing demand. So over the next 30 minutes or so, we are going to be primarily focusing on fish oil as a resource and strategies to try to replace that particular ingredient. Why? Well, that's because fish oil remains the only economical source of long-chain polyunsaturated fatty acids, or as you'll see it referred to in this presentation, LC-PUFA. Those, of course, include arachidonic acid. You'll see that shown as ARA or 24N6 in this presentation. Eicosapentaenoic acid, EPA, or 25N3, and docosahexaenoic acid, or DHA, 22,6N3. And again, you're going to hear and see those acronyms and numeric abbreviations used throughout this presentation. LC-PUFA have critical physiological roles in vertebrates, and they are essential nutrients that must be consumed directly by at least some species of farmed fish. So we need to think of these nutrients from a basic nutrition perspective when we're formulating feeds. But of course, LC-PUFA are also really important in terms of human health and well-being. They are what makes seafood uniquely nutritious in the human diet. So as we'll see later, when you reduce levels of LC-PUFA in the feed, you're also typically reducing levels of those same nutrients in the edible tissues of fish. So when we're formulating feeds, we really need to think about first the fatty acid requirements of the target species, but also the expectations of the consumers that will ultimately eat that fish. So nutritionists have already done considerable work to spare fish oil in aquaculture feeds. I'm sure many of you are familiar with a lot of that literature. And you can use a wide variety of terrestrial origin fats and oils. Generally speaking, and this is just a very rough rule of thumb, but you can typically replace as much as half of the fish oil in a formulation with pretty much any other type of alternative lipid without affecting growth. But for a lot of species, further reductions or completely replacing fish oil with any type of alternative lipid source has proven very difficult, and it typically results in impaired production performance. So if you want to play a game, if you want to play the game and win, it really helps to know the rules. So for further progress in fish oil sparing, that means that we need to better understand fatty acid requirements, those rules, and the demands and tolerances of farmed fish. So to tell this story, I'm going to review a few studies from some other labs, but most of what you're going to be seeing here is our group's publications on the subjects that we're talking about here today. And I will note that, sure, my name is on the publication, but this is a lot of students and postdocs and technicians that have really brought this work to fruition. So credit to all of them. So before we delve further into the rules of the lipid nutrition game and fatty acid requirements in fish, let's review what molecules we're actually talking about. This figure shows the biosynthetic pathways from 18O, stearic acid, a saturated fatty acid, to the N3 and N6 long-chain polyunsaturated fatty acids. The first step is transforming 18O into 18-1 and 9 through the action of the delta-9 desaturase. That's easy enough to do. But the next step, transforming 18-1 and 9 into 18-2 and 6 or into 18-3 and 3, that's where we first run into trouble. These transformations and the production of those two C18-PUFA, as you'll see, I refer to them later, those are not known to occur in any vertebrate, and that includes fish. But if you start with either of those two C18-PUFA, from each through a series of elongation and desaturation steps, 18-2 and 6 and 18-3 and 3 can be transformed into those LC-PUFA we mentioned before, ARA, EPA, and DHA. Those are the ones I'm showing here in purple. These transformations are made by some fish, but not by other fish, or at least not all species are able to biosynthesize LC-PUFA from those 18 carbon precursors in appreciable amounts. Now, because 18-2 and 6 and 18-3 and 3 cannot be synthesized, they are considered to be essential fatty acids in most cases. Now, for those species which cannot transform these into ARA, EPA, or DHA, it's those long-chain PUFA that are also considered essential. And this is fine, and this is correct in terms of how we typically define nutrients as being either essential or expendable. In essence, the question is, do we need them? And if so, can we synthesize them on our own or not? However, I think it's really important to remind ourselves that the only reason that 18.2 and 6 and 18.3 and 3 are considered essential is because we can use them to make something else, specifically the LC PUFA that have these distinct physiological functions. The 18 carbon precursors don't really seem to have any physiological function other than to serve as precursors for the LC PUFA that do have these important roles. Roles like maintaining membrane confidence and fluidity, or as precursors to regulatory compounds like eicosanoids, docosanoids, and others. Now, for those species that cannot produce LC PUFA de novo, it's really only those fatty acids that are essential. We shouldn't say that those fish require the LC PUFA and the C18 PUFA precursors. They either require one type of fatty acid or they require the other. Now, for those species that can produce LC PUFA de novo, they obviously have options. Their PUFA demands can be met either by providing the precursors or by providing the LC PUFA themselves. But in the strictest sense, it's only the precursors that would be considered essential in that case. But again, let's not forget what the fish needs. At the end of the day, it's really those LC PUFA that are either provided directly or indirectly. Now, a number of studies have been conducted to determine the essential fatty acid requirements of fish, and I'm going to begin by a very brief review of a few cool and cold water taxa. So here at the top, we have rainbow trout. Reportedly, the N3 fatty acid requirements of this fish can be met by providing 18.3N3 or N3-LC PUFA, specifically EPA and DHA, in the diet. Pacific salmonids needs can reportedly be met through the provision of just those 18 carbon PUFA precursors. Other fish, however, are not so amenable. This includes the various Moroni species and their hybrids, which are generally reported to require intact LC PUFA in the diet, so they can't use the C18 PUFA precursors. Okay, that's cool and cold water taxa. Let's take a look at warm water taxa, just a few. There's also some variation here. Tilapias and catfishes are relatively flexible. Like the rainbow trout, like the Pacific salmonids, they can apparently utilize 18 carbon PUFA or LC PUFA. They're not so picky. Other warm water species, however, like largemouth bass and cobia, are not as flexible, and they appear to require at least DHA intact in the diet and potentially other LC PUFA. Now, I often hear people cite this old conventional guidance that cold water species require LC PUFA in the diet, whereas warm water species do not. Well, it really didn't take more than a handful of species being reviewed to overturn that convention. There are some cold water species that do not require LC PUFA, and there are certainly warm water species that do. Now, of course, there are exceptions to every rule, but I think that there are enough exceptions to this cold water, warm water rule to basically render it almost useless. Now let's explore this conventional wisdom that's related to fatty acid requirements a little bit more. In addition to this cold water, warm water dichotomy, some people cite the salinity dichotomy, which suggests that marine fish require LC PUFA, whereas freshwater fish do not. Now in this table, I've compiled a number of different species to represent cold, cool, and warm water fish, as well as species to represent marine, freshwater, or euryhaline species. I've pulled their basic essential fatty acid requirements from the literature. In short, do they require intact LC PUFA in the diet or not? Now, our group spent a lot of time looking at this table. We were wrestling with this information. We really were struggling to make sense of it, as these old conventional rules based on temperature and salinity preferences really didn't seem to be holding up in very many circumstances. So to try to make sense of some of the published requirements for either C18 PUFA or LC PUFA, I decided to look at the information in a little different way. What I'm showing you in this figure are the same species from the previous table, but I've grouped them vertically by their salinity guild, and I've color-coded them to reflect their temperature guild. So the warm water species have those red vertical lines, the cool water species are in light blue, and the cold water species are in dark blue. But the difference here is that I've also ranked them all along the x-axis by their trophic level. Trophic levels are numeric rankings given to organisms based on what they eat. Primary producers get a value of 1, primary consumers that eat number 1s get a value of 2, consumers that eat number 2s get a value of 3, and so on, up to a maximum level of 5, which is meant to represent the apex predators in the ecosystem. So you just figure out what an animal is typically eating in the wild, and based on what it's eating and in what proportions, you can calculate this average trophic level ranking, which has been done for thousands of fish species throughout the world. Now let's overlay on this figure those LC PUFA requirements and see how things shake out. As it happens, each of the species that are highlighted here in the purple overlay require LC PUFA. None of those that are in the yellow overlay do. As you can see, there's no rhyme or reason to this based on temperature or salinity preference. Each group is represented at least once in each group. The only thing that really matters here in this figure is trophic level, with organisms feeding above a certain point on the food chain requiring intact LC PUFA, and the rest being able to transform those 18 carbon precursors into those physiologically relevant molecules that they need. Now, as you can imagine, after struggling with those tables for so long and trying to make sense of them, this was an immensely satisfying finding. We thought we really had a new rule that might create order from the chaos, but this wasn't a hypothesis that had been thoroughly tested. So we created a series of experiments to really try to go beyond the previous work to define fatty acid requirements, which, number one, typically have ignored the N6 PUFA. Remember, those nutrients have a role to play here too. And most of the historical work to this point didn't really adequately differentiate in most cases between the different types of N3 PUFA. What I mean by that is that a lot of the previous work often defined fatty acid requirements in terms of a total N3 fatty acids requirement, including both the C18 PUFA and the LC PUFA. Or sometimes those requirements would be defined as a total of EPA and DHA. In fact, those that are familiar with the 2011 NRC nutrient requirements of fish and shrimp will recognize those groupings from the nutrient requirement tables. That's how fatty acid requirements are reported in that particular text. These nutrients are biochemically related, but they're not interchangeable, at least not in all species, and they do fulfill different physiological functions. These experiments that I'm about to share with you were intended to examine the essentiality of N3 and N6 PUFA as unique nutrients and to really test our hypothesis of LC PUFA essentiality as a function of trophic level in fish. So we ran a series of trials with hybrid striped bass, channel catfish, Nile tilapia, Florida pompano, and rainbow trout that were fed diets containing fish oil, hydrogenated soybean oil that is completely devoid of PUFA, or that same hydrogenated soybean oil supplemented with 18,3-N3, 18,3-N3 and 18,2-N6, DHA, ARA and DHA, or all three of the LC PUFA that we're concerned with, ARA, EPA, and DHA. What we learned was that LC PUFA were clearly essential in the diets of high trophic level species carnivores like hybrid striped bass. Mid-level carnivores like Florida pompano and rainbow trout could certainly utilize C18 PUFA to meet their essential fatty acid requirements, but providing those intact LC PUFA seemed to provide some advantage, most likely by precluding the need for those fish to expend energy. They just had an energy savings and were able to put that energy into additional growth. Providing low trophic level species, herbivores or omnivores like tilapia and channel catfish with those LC PUFA in the diet did not have any positive effect on growth or other performance metrics. In fact, fortifying diets with EPA and DHA appeared to induce a rather insidious 24-N6 deficiency. And to the far right on this slide are two figures that are going to illustrate this point in tilapia specifically. 23N9 and 22,5-N6 are relatively obscure fatty acids that represent dead ends in the biosynthetic pathways for 24N6 ARA and 22,6N3 DHA. An increase in the ratio of 23N9 to 24N6 indicates a deficiency in 24N6. In other words, the accumulation of 23N9 in the tissues is indicative of an animal that is trying but ultimately failing to synthesize ARA. And it's indicative of a developing N6 PUFA deficiency. An increase in the ratio of 22,5-N6 to 22,6N3 is similarly indicative of a developing N3 PUFA deficiency. Now, we would expect to see elevated deficiency ratios in fish that were fed the essential fatty acid-free negative control that's shown here in white. So there's no surprise there. We don't see elevated N3 deficiency ratios in the bottom panel in any of the other treatment groups because they all contain some DHA or ALA from which the tilapia can produce the DHA that they need. What is interesting is that we see in the top panel N6 PUFA deficiency among tilapia that were fed the essential fatty acid-free control, as well as the diets that contain an abundance of N3 LC PUFA, specifically the DHA and LC PUFA diets that are shown here in light gold and in purple. It's well known that increasing consumption of LC-PUFA will downregulate the activity of delta-5 and delta-6 desaturase and ultimately downregulate de novo synthesis of LC-PUFA. I mean, this makes sense. Why would you bother producing LC-PUFA yourself and expending energy if you're already consuming more than enough of those nutrients? But what appears to be happening here is that the overabundance of N3-LC-PUFA is telling the fish to downregulate delta-5 and delta-6, which also shuts down N6-LC-PUFA synthesis. The result is that the fish are biochemically hobbled, and they're unable to satisfy this demand for ARA when they are otherwise perfectly capable of doing so. Many papers caution about the importance of formulating diets to contain a proper balance of N3 and N6 PUFA. But this work was really, I think, unique in that it demonstrated the consequences of failing to heed that advice and explained why you often see growth suppression in these cases when you have an N3 and 6 PUFA imbalance. But perhaps more importantly, this collection of studies helped to populate the figure that I'm showing here and really firmly establish this paradigm of using trophic levels to predict C18 PUFA versus LC PUFA essentiality in fish. This figure is more elaborate, of course, than the one I showed you before, and it doesn't have the cute fish pictures on it, but the organization is basically the same. It's just now that we have close to 40 fish species that are numerically coded. They've been grouped along the y-axis in terms of their salinity preference. They're color-coded, again, to reflect their temperature preferences, and again, they're ranked along the x-axis according to their trophic level. The white dots denote the species that do not require LC PUFA. The black dots denote those that do, and you'll see there's a handful of yellow dots here that represent studies that either had an incomplete experimental design or ultimately yielded inconclusive results. What you see is a quite clear separation between the low trophic level species that can get by with C18 PUFA, the high trophic level species at the other end of the spectrum that cannot, and then those species with middling trophic levels having somewhat middling results, which is consistent with the idea that while not strictly essential, providing LC PUFA to those species in the middle can provide some energetic advantage. All of the results of this work were published together in a special section of the North American Journal of Aquaculture earlier this year. And in their introduction to that work, the guest editors, David Francis and Monica Betancourt, stated that, quote, these studies chart a far more reliable mechanism for predicting the essential fatty acid requirements of fish species, offering a highly valuable and insightful contribution to the field of fish nutrition as it continues to delve deeper into marine resource substitution. So again, going back to what Albert said about, in the research community, we're here to provide solutions. This is very gratifying to be able to put this information out into the public sphere and say, here is a tool, here's something that you can use. You can rely on these trophic levels to help you predict and understand ultimately what an optimal feed should look like for those species, including those for which we'll never have an opportunity to do quantitative nutrient requirement studies. So, with that, we've established a new rule that trophic level is a better predictor of C18 versus LC PUFA essentiality in fishes. This is an important advance in our understanding. Again, there are many hundreds of species that are being cultured around the world, and we're never going to be able to do comprehensive requirements in all of them. So that's a really important first step. But for those species that do require LC PUFA, now let's ask ourselves the question whether all of those LC PUFA are equally important in terms of satisfying dietary demand. In our early fish oil sparing work with cobia, we found that complete replacement resulted in reduced growth and reduced survival. Cobia are a highly predatory fish, so given what we've just discussed about trophic levels, it really shouldn't come as a surprise that they require intact LC PUFA in the diet. But among the N3 LC PUFA, do they require them equally? Are EPA and DHA both essential in their diets? To answer this question and to find a way around the roadblock that we had encountered during our fish oil sparing work, we needed to define their requirements a bit more explicitly. So we designed an experiment in which juvenile cobia were fed diets containing fish oil, soybean oil, or soybean oil that was amended with EPA, DHA, or both of those fatty acids at levels representing either 50% or 100% of the levels that were observed in the fish oil control feed. So what happened? Well, as predicted, the fish oil control feed that's shown here in black yielded good weight gain, that's in the upper panel, and a reasonably low FCR, as you can see in the lower panel. The soy oil control in white yielded the opposite, with meager weight gain and elevated FCRs. What happens when we add EPA to the diet? Well, the short answer is pretty much nothing. The weight gains stay low and the FCRs stay high, as you can see here in those green bars. But when you add DHA at either 50% or 100% of the supplementation levels, both weight gain and FCR are restored. And you get about the same effect when you supplement with EPA and DHA simultaneously. What we concluded from this study was that DHA is crucial and truly essential in diets for cobia, but EPA is largely dispensable. This isn't to say that EPA doesn't have a physiological function in cobia. EPA plays important roles in all vertebrates, and there's no reason to suspect that cobia are unique in that regard. It's just that EPA is probably needed at much lower levels, and what little demand is there, that might actually be met through retroconversion of DHA back to EPA. And I think that's another minor but potentially important rule. Although there are species that cannot synthesize forward and turn EPA into DHA, some of them can synthesize backward and turn DHA that they consume in the diet into EPA de novo. Now, as you might imagine, there was a little bit of pushback when we first presented these data, but we have since documented similar findings in other carnivorous fish species, including California yellowtail and the omnipresent Atlantic salmon. So now we've got a couple of additional insights that we can use to our advantage. We know the part about trophic levels, and we also know that not all LC PUFA are created equal, both of which can give us considerable flexibility in formulating to meet the nutrient requirements of our target species. But as I mentioned at the beginning, we really need to think beyond just meeting the fish's requirements and consider how LC PUFA are ultimately deposited in the edible tissues of fish. Now here's a slide that I put together showing the relationship between dietary DHA content on the x-axis and fillet DHA content for a number of different fish species on the y-axis. The first thing to note here is that all species bioaccumulate DHA in their tissues. Nearly all of the points fall above that dashed line that represents a one-to-one relationship. So fillet levels are almost always higher relative to what the fish is consuming. They're all accumulating DHA to a certain extent, but why is there so much variability here if this really is a kind of fish are what they eat sort of relationship? Well, as with the requirements, we can dispense with the thermal and salinity guilds right away. Here I'm highlighting the freshwater species with circles, the marine species with triangles, and the temperature guilds according to those same color coding conventions that I've used elsewhere in this presentation. And as you can see, there really aren't any patterns or groupings that are jumping out here. Interestingly, the ability of a fish to produce DHA de novo doesn't really explain much either. Although that you might expect fish that are capable of producing their own DHA, those are shown here on white, might deposit more in their fillets than those that rely exclusively on what they're fed. Those are the ones that are shown in gray. There really aren't any clear patterns here to suggest that that's true. So to see what's driving this variation, we're going to focus on one particularly variable data set, which involved white sea bass. Each of these points represents juvenile white sea bass that were fed different dietary levels of DHA. But again, what is driving this variation? Some of our previous research in other species suggested that alternative lipids that are rich in saturated fatty acids had counterintuitive effects on the composition of fish tissues. Whereas fish that were fed alternative lipids like soy and canola grew to resemble their diets and contained very high proportions of C18 PUFA, fish that were fed alternative lipids like palm that were rich in saturated fatty acids didn't have high levels of those fatty acids in their tissues. In fact, the composition of their tissues was very strikingly similar to that of fish that were just fed fish oil. So to explore this phenomenon further, we set out and designed this experiment to feed white sea bass diets that contained fish oil or rated levels of standard soybean oil containing mostly 18.2 and 6, or hydrogenated soybean oil containing mostly 18O. Now, when you hydrogenate a lipid, any polyunsaturates that are present are first turned into monounsaturates, and then those monounsaturates are biochemically turned into saturates. So regular old soybean oil has a lot of 18.2 and 6. That gets turned into 18.1 and 9, and then ultimately the 18.1 and 9 gets turned into 18O. So as we replaced fish oil with more and more traditional soybean oil, the diets contained increasing amounts of 18.2 and 6 and a corresponding decline in EPA and DHA. With a hydrogenated soybean oil, dietary EPA and DHA are also declining by the same amount, but in this case, they were replaced by 18O. What we observed in this case was that growth was reduced among fish that were fed those very high levels of traditional soybean oil. No surprise there, that was consistent with the information that was available at the time for white sea bass and our previous experiences with other marine carnivores. But what was shocking is that we didn't really see any growth suppression among fish that were fed high levels of the hydrogenated soybean oil. We also saw very little effect on the hydrogenated soybean oil on tissue composition. So whereas on the other hand, we observed very dramatic changes in tissue fatty acid profile among those fish that were fed the traditional 18-2-6 rich soybean oil. And that's what this figure here is illustrating. It shows the relationship between dietary DHA levels and fillet DHA levels for the fish that are fed increasing amounts of either traditional soybean oil, those green colors, and hydrogenated soybean oil in the purples. And I'll just point out that the single black dot there, that represents the fillet composition of fish that were fed the fish oil control diet. Fish that are fed the regular soybean oil soybean oil diets accumulate DHA in a typical fashion. When they aren't fed much DHA, their tissues don't contain very much DHA. But for the fish that are fed hydrogenated soybean oil, they're maintaining almost the same levels of DHA as the fish oil control, despite being fed much, much less DHA. From this, we concluded that alternative lipids containing high levels of saturated fatty acids have a disproportionately small effect on growth and on tissue composition, and that can facilitate much greater levels of fish oil sparing than a traditional C18 PUFA-rich alternative lipid would allow. Again, these results were pretty puzzling to quite a few folks when they first came out, but the LC-PUFA sparing effect, as it's now been called, has been documented by our group and many others in many different species in the intervening years. And just to really drive this point home a little bit further, I'll note that it's not just tissue deposition of DHA or LC-PUFA that is affected by these dietary levels of saturates versus C18 PUFA. It's really the whole fillet profile. I'll just take a moment here to orient everyone to these figures. On both of these radial diagrams, we have the major fatty acids and fatty acid groupings from the fillets. Starting at the top and moving in a clockwise fashion, we are increasing in chain length and in the degree of unsaturation. So at the top, we have axes for the saturates, moving into the monounsaturates and the C18 PUFA, or the MC PUFA, medium chain PUFA, as they are called here, and then finally into the LC PUFA. The green lines represent fillet composition of fish that were fed diets containing increasing amounts of the standard C18 PUFA-rich soybean oil relative to the composition of fish that were fed the fish oil control feed, which is the one that's represented by that heavy black circle in the center. Now, because we're using ratios, points that fall outside of that circle represent higher levels of a particular fatty acid relative to control, whereas points inside the circle represent lower levels. So again, the figure on the left is for the composition of fish that were fed the C18 PUFA-rich soybean oil, and the figure on the right is the same thing, only for those that were fed the hydrogenated, saturated fatty acid-rich soybean oil diets. The more fish oil you replace with either alternative lipid, the greater the degree of fillet profile distortion you see. That's represented by greater and greater deviations from that perfect circle. And that's certainly true for both types of soybean oil. But perhaps the most important takeaway here is that fish that are fed the saturated fatty acid-rich soy don't exhibit nearly the same degree of profile modification, and they maintain an equivalent level of almost all of the different fatty acids that we examined. More importantly, they maintain tissue levels of LC-PUFA, and they do not accumulate saturated fatty acids, despite consuming substantially less LC-PUFA and vastly more saturates. So while the conventional wisdom is that fish are what they eat when it comes to fatty acids, when it comes to saturated fatty acids, that really isn't the case. And this is an important exception to the rule that we can exploit to our benefit when formulating reduced fish oil feeds. And as I alluded to before, not only does saturated fatty acid content in the diet influence tissue deposition, it also appears to influence LC-PUFA requirements. These two figures illustrate the growth performance of white sea bass that were fed the diets I mentioned before. When you decrease dietary LC-PUFA levels in the standard soybean oil diet below a certain point, you do see a growth performance drop. However, that same breakpoint does not appear among the fish that are fed the saturated fatty acid-rich hydrogenated soybean oil feeds. And that suggests that when it's provided in the context of saturated fatty acid-rich feeds, the LC-PUFA demand of white sea bass is actually lower than it is in the context of that standard soybean oil diet. We've seen this effect in white sea bass as well as a number of other fishes. Collectively, these data suggest that we need to start thinking of essential fatty acid requirements, not in terms of absolutes, but in terms of plastic interactions with other dietary constituents, which may be driving LC-PUFA requirements up or down. Now, that's not especially novel thinking. We already consider these kinds of interactions for certain amino acids, for antioxidants, and so forth, but it is a fairly novel idea for lipid nutrition. And I think our work really kind of helped lay the foundation for this new perspective and a new approach to fish oil sparing. So a few concluding remarks to really summarize the research I've shared with you. First, essential fatty acid requirements must be defined in terms of specific fatty acids based on conserved physiological functions. Not only do we need to stop thinking of 18-2 and 6 and 18-3 and 3 as anything other than a means to an end, we also need to think of requirements in terms of individual fatty acids. In other words, an ARA requirement, a DHA requirement, not a total LC-PUFA requirement or a total N3 fatty acid requirement. Remember that requirements for LC-PUFA are based on the inability to synthesize those nutrients de novo, but that ultimately what all fish need are those LC-PUFA, not the precursor C18 C18-PUFA precursors. If we are to make generalizations about the kinds of fish that require LC-PUFA and the kinds of fish that don't, we should recognize that trophic level is a much more instructive criterion than salinity or temperature preference. And I will note here that while we think of that as maybe being kind of a big takeaway from all this research, I'll note that there is nothing new under the sun. It might be new to us, but that very concept, using trophic level and separating fish based on carnivory versus herbivory, that was something that was described in the literature by Sargent and his colleagues almost 25 years ago. Maybe we have more proof now, maybe we have more evidence now, but this is not necessarily a completely new earth-shattering idea. It's just something we need to do a better job of applying. And finally, we all recognize that the essential fatty acid requirements of fish species are likely to vary among life stages and rearing conditions, but we also need to start thinking about the influence of dietary composition, particularly saturated fatty acid content, as another driver of essential fatty acid requirements, one that is under our control and we may be able to use to our strategic advantage when we're pursuing fish oil sparing and optimal nutrition of cultured fishes. As I mentioned earlier, our PUFA essentiality studies and a broader synthesis of our work in lipid nutrition was published earlier this year in this special section of the North American Journal of Aquaculture. This was really kind of something of a magnum opus for me and my work as a nutritionist. So it was a great moment to see this go out into the literature, and thank you to the guest editors and of course to all the contributors who helped make that work happen. I'd like to pivot just briefly and spend a few minutes talking about the future of fish nutrition. I believe that sustainability is really rooted in judicious use of a wide variety of feed ingredients. Reduction fisheries existed before aquaculture became such a dominant sector, and those fisheries will continue to exist with or without demand from our industry. Fish meal and fish oil can be used in poultry feeds and pet diets, but the aquaculture industry can do a lot more with those resources than other industries can. Fish meal and fish oil still have a role to play in aquaculture, and these resources can be used sustainably, in my opinion. But of course, the inclusion rates are what we have to consider. As we explore alternative sources of protein, lipid, and other nutrients, we need to be mindful of both the ecological and economic sustainability of those feeds. It's my view that with strategic R&D, aquaculture feeds and the aquaculture sector can achieve both of those benchmarks. But a certain degree of realignment in the aquaculture nutrition research community is needed to fully achieve those goals. And this is a subject that, that I explored with Gio Turchini and Brett Glencross. I think at least one of those gentlemen is on the call now. So shout out to my guys. We explored these ideas in a thought piece that we published last year, and I'm actually going to highlight some of these points just in my few minutes that are remaining. If our goal is to help industry reduce its use of fish meal and fish oil, we need to think about it a little bit like a fish farmer would. The alternative ingredients must meet certain benchmarks, and I've listed those here. They're not revolutionary, but they are important characteristics for all of us to consider, particularly the researchers who maybe aren't dealing with all of these variables all the time. First and foremost, the feedstuffs must support rapid and efficient growth. But more than that, they must provide everything that the fish need to thrive. I think we all recognize the limitations of approaching animal feeding from the perspective of basic nutrition. Alternative formulations must go beyond basic nutrition. They must provide the right balance of essential, conditionally essential, and non-essential nutrients that are needed for fish to fully realize their bioenergetic potential. They must also yield products that meet our and our customers' expectations for taste, nutritional value, safety, and other factors. Aquaculture can't accomplish the good that we hope to with underwhelming seafood that doesn't taste very good. And further to that point, farmers won't be in business to do that good work if the feeds aren't cost effective. We recognize that cheap feeds are sometimes just that, and that quality nutrition often comes with a higher price tag. But the feeds still have to be economical, not just for the financial health of aquaculture businesses, but for their customers too. Theoretically, an aquaculture business can realize the exact same profit margins with more and more expensive feed inputs if they're willing to pass those increasing costs on to their customers. Of course, this would require a customer base that is not only able, but willing to pay a higher and higher price. But I think even the Whole Foods shoppers of the world and the other kind of high-end markets, their customers have a limit to what they're willing to pay. Even their customers have a limit. So the aquaculture industry should really strive to put superior seafood within everyone's reach, and we have to have cost-effective feeds to get there. Least cost feed formulation works really best when you have a diverse portfolio of ingredients to work with. Those ingredients have to have known nutrient profiles and digestibilities, and they have to have consistent composition, or at the very least, they need to vary predictably in their nutrient composition. They must be consistently available in volumes that are relevant to the scale of our industry. Now, that appears to be changing for products like black soldier fly, like I'm showing here, but that has been a really long time coming, and supplies of those ingredients are still below perhaps what demand would actually be for those ingredients. So we need to kind of expand not only the portfolio, but the volumes of those ingredients that are available. Of course, the ingredients cannot contain unacceptable levels of contaminants or anti-nutritional factors. That's another variable or suite of variables for us to consider. And the ingredients must not lead to negative shifts in nutrient retention or our ability to capture waste. Digestibility is a part of this, of course, but it also has to do with how the animals react to the ingredient and how it affects their gut transit times, fecal stability, etc. And once again, the pricing needs to be competitive. I think much of the data to support the development of new ingredients is focused on just those ingredients, and I think that we need to try to not do this work in a vacuum. We need to stop thinking of ingredients as just substitutes for fish meal and fish oil. New ingredients don't just compete with fish meal and fish oil, they compete with everything else that competes with fish meal and fish oil. We need to think about an ingredient's functional and physical properties, and we also need to think about how those attributes how those attributes are in terms of the overall mix, and how certain ingredients are going to play together in the context of a formulation. And so to those ends, here are a few objectives that I would suggest to really address what I see as critical needs and knowledge gaps in aquaculture nutrition. I would recommend that we expand globally the availability of open source nutrient digestibility databases as industry resources. There's a handful of these that are available, and they are really critical resources. And so investing in those and keeping those things up to date, I think, is really important. I would also suggest that it would be possible to, or beneficial to develop a similar resource that describes the physical properties of ingredients and recommended extrusion practices. I think that would be really beneficial to the industry. Although we in the nutrition community, we spent decades developing a really substantial body of knowledge related to nutrient composition and biological responses to different feeds and different ingredients, comparatively little attention has been paid to the manufacturing processes. We recognize that manufacturing factors can influence nutrient availability and those biological responses in our fish, but we kind of acknowledge this in a relatively perfunctory manner. We just say that, well, extrusion is generally better than steam pelleting, but we really haven't done as much as we could do, in my view, to support the industry as it looks to incorporate these new ingredients into existing practices. I don't think that we've tapped into the considerable amount of innovation that has occurred in the field of extrusion science, both for animal feeds and human foods. I think that's an area that's really ripe for exploitation. I think it would be wonderful to see more of our researchers exploiting kind of a crowdsourced model. So if your particular lab has a lab scale extruder and you can make experimental feeds for lots of other people in your immediate area and help them expand their ability to do research, I think that's a wonderful thing to do. Or if your lab has a particular piece of analytical equipment that allows you to assess something in the feed or something in the fish that maybe not a lot of other people are able to do, set up that collaborative network so that we can work together and see that rising tide lift all ships. As I said before, I think there's a lot to be gained from exploring these alternative manufacturing technologies and really bringing a stronger focus on not just the nutritional aspects of it, of feed production, but also the extrusion science elements of it as well. And finally, I would encourage those in the nutrition research community to really fully engage in the regulatory approval process for new products that are being developed. I know that sometimes regulatory science isn't very sexy. It's maybe not always a lot of fun compared to a lot of the really novel work that's being done around the world. But we have to remember that this is where we can contribute directly to innovation and technology transfer in industry. We can do really great science to understand nutritional physiology and prove the value of new ingredients. But if they can't be used by industry, then what have we really accomplished? And so in my own mind, I think back to that 20-something-year-old student that was in college and wondering what she was going to do with the rest of her life. And I think, okay, what would she have to say about this? And I think she would say, let's do everything that we have to do in order to get these tools, put these resources in the hands of the industry that we support so much. So with that, I'll say thank you and apologies for going over time, but hopefully there's a bit of time left for questions.
I think we have, thank you, JC, so much. I think we have ample time if we can keep the audience alert, hopefully. But before we go to the questions, I would like to also thank Jeffo Nutrition Inc. to allow us to and give the opportunity to conduct these sessions, giving us the platform. And Jeffo is kind of a 38-year-old company, started with I would say a visionary, because the person, Mr. John Fontaine, is president of Jeffo. He just graduated from the university with a degree in agronomy, and then tried to think to help the farmers around the small town in Quebec. And that's how it started, right? Carrying the bags of products to the farmers' places, just to help farmers. And that is our objective, to help the producers and farmers. I think that's why we are here today. So thank you, Jeffo, to give us this platform and opportunity. Before we start, we have a lot of questions, not a lot, but quite a few questions. Before we start, I would like to read one statement. It's for Jesse from Dr. Tutini. No questions from me, but I simply wanted to say thank you. These webinars are great, and listening to Jesse was very informative. And what a champion, end quote. This made me think that COVID actually did something good. It made the world smaller, okay? And we can now more easily connect and enjoy top quality scientific talks more frequently, end quote, without the need to travel around the world for conferences and symposia, end quote. So thank you and well done, okay?
Thank you, and thank you to Gio. That's very, very kind.
Yeah. So I'll start with a very simple question that what would be the standard ratio for SFA and PUFA in the diet that you said, 50-50? I think that's where you said they get the best results, and then you have.
Yeah, I mean, of course, it really depends. That's the scientific answer, right? If you ask a scientist anything, the answer is always, well, it depends. It depends on the degree of fish oil sparing that you're trying to achieve and so forth. But I think kind of as a general rule, what we tried to match up with in the diet was, we looked at, okay, what's the composition of fish oil? right or wrong, that is the gold standard that we always compare ourselves to. So what are the rough proportions of saturates, monounsaturates, and polyunsaturates in fish oil? Understanding that most of the polyunsaturates that are there are in the long chain variety, not in the C18 PUFA variety. So if we are trying to replace fish oil, if we replace it with one of those soy or canola types of ingredients, we're really disrupting that saturate, monounsaturate, polyunsaturate ratio, and we're overloading it with those C18 PUFA. But with using saturated lipids or even terrestrial rendered animal products that have a lot of monounsaturates in it, you can get a very similar effect by trying to kind of match that balance. So for fish oil, it's roughly a third, a third, a third. A third saturates, a third monounsaturates, and a third polyunsaturates. So I think we had a lot of our greatest success trying to match that.
Awesome. So related to this, there was one question from, I think that's from Alejandro D'Amato. This is a interesting question. It's like about finding a new oil source with high in saturated fat. So what do you think about using the calcium salts that are used in dairy cattle? Like it's a bypass palm oil, right? Palm fat, high in saturated fat. And there, I know there are some studies done in Malaysia with Dr. Wing Kiong, by Dr. Wing Kiong, right, on Malaysian palm oil in this area. And what is your opinion on that?
Yeah, I think that's a really interesting question. I think my first reaction to that would probably be kind of similar to the question that we got a lot when we first started just looking at some of the kind of harder saturates. And that's okay. So what's our digestibility limitation going to be on some of those things? Because with some of those calcium salts, that's the whole point, right? Is that they're really resistant to digestion to get them, past the rumen and so forth. So I guess that would be my first question. But yeah, I think that's a fascinating idea, and potentially, maybe another resource that we haven't tapped into, another ingredient resource, I should say.
Good, thank you so much. The next question is from, I think I answered this one. I answered this one. From Mr. Arifin Rahman, I think he is doing PhD in Thailand, I think some university. So the question is there any relationship between dietary fatty acid and serum protein? And I found 2 experiments, lower serum protein in Asian sea bass when fish oil is completely replaced by vegetable oil or blend of vegetable oil. Any idea on that? Or any comments?
So yeah, kind of shooting from the hip here because we didn't do a lot of work to look at serum proteins. Actually, I'm trying to think back through the archives. I think we really only did one study where we were looking at the composition of the lipoprotein fraction. So we were just looking at the fatty acid profile of what's circulating in the bloodstream, and unsurprisingly, what's in the diet does have an influence on what's circulating in the bloodstream. But I don't think we actually looked at the actual amounts of serum protein, if that's the question. But I think it's reasonable to expect that there could be some influence, right? We know that the types of fats that we consume as humans influences the composition of our triglyceride levels and cholesterol levels and so forth. So I think in that case, and probably in the case of fish, it has less to do with the kind of fatty acid, and maybe more so to do with the form of presentation. So is this a phospholipid? Is this triglyceride? Is it, what exactly are you feeding your animals? I think that's probably more likely to have an influence. The lipid class maybe would have a greater influence than the fatty acid composition specifically. But yeah, who knows? Somebody better do the study and find out.
Now coming to some, I think it is kind of a new area of work that we have been doing about nutritional programming, right? It's been happening from France. Quite a few papers came out recently. So intake of low HUFA diet at first feeding improves, I think, the ability of juveniles, right, to synthesize more LC-PUFA. Any new information on this? And
Yeah, I think this concept of nutritional programming, or even like looking at the maternal influence and how that's ultimately going to determine the trajectory of the offspring, not even from a genetic perspective, but just the kind of epigenetic effect of what you're feeding mom, and that's influencing the trajectory of the fish and what they're capable of physiologically. I guess I think about this stuff from a couple of different perspectives. So, you know, obviously from a nutrition perspective, but you know, Riverence is first and foremost a breeding company. So yes, we do raise a lot of rainbow trout. We're one of the largest producers of farm trout in the world. But we started and continue to be a breeding company. So we think about these things. We think about how we might be able to kind of program our offspring to be better in one way, shape, or form or another. And certainly, you know, having a fish that's able to produce more long chain pupa given the same amount of dietary inputs is advantageous. But I think you always have to balance that with just because a fish can do something doesn't mean that it will. And what I mean by this is, if you look back to a lot of the literature from, say, the early 2000s, and maybe,
Right.
well, certainly mid 2000s to this point, there was a lot of work that was being done to say, well, okay, we really want more EPA and DHA in the fillets, so we're going to load up the diet with a lot of 18-3 and 3. We're going to push as many of the precursors as we can. And yeah, you can technically see an upregulation of LC pupa synthesis in those species that can do that. They will produce a little bit more, but they won't come anywhere close to what they would have in their tissues if you just fed them the fish oil directly. So then, well, that wasn't working out how we wanted it to. So, well, let's try to skip a step. Let's look at some of these stearidonic acid lipid sources. So now it's not 18-3 and 3, now it's 18-4 and 3. So we think we skipped the first rate limiting step. The problem is that you just run into the next rate limiting step.
Right,
and
right.
yes, the fish are going to produce a little bit more than they would have otherwise, but they're still not going to achieve those levels. And like I said, the fish can do it, but they're only going to produce those LC
So
pupa
much.
to the extent that they need it, right?
Right.
Nobody told them what our expectations are as far as, we really want this in the fillet, so if you could hurry up and synthesize that, that'd be great. So, you know, there's always going to be that biochemical limitation. So that's my only caution for work like that is, you know, are you producing a fish that is, I guess, kind of energetically making bad decisions, that they're choosing to produce more of these fatty acids. It's really not a process that, from a biochemical, bioenergetic perspective, they shouldn't want to expend extra energy doing that. And as with any kind of breeding selections, when you're selecting for one thing, you are always inadvertently selecting against something else or for something else that may or may not be of value to you. So I think just approach it cautiously, but I think it's a really interesting area of work.
So what is your opinion about if we want to say enrich tilapia fillet with LC pupa, significant amount, and you know that could be a good for human health marketing perspective, and then new kind
Sure.
of product, right? Like omega-3 eggs or DHA milk, right?
Or peanut butter.
Yeah.
Yeah. Yeah. So I think, you know, especially with tilapia, I think you're going to run up against kind of the natural biological limits of what those animals are going to do. You know, obviously tilapia are a very
Right.
lean fleshed fish, so there's only so much lipid you can pack into those fillets.
Right.
But I also think, sometimes I think of tilapia as being kind of like, they're obviously not a ruminant, but they're the closest thing to a cow we have in fish, right? So I think the way that they metabolize fatty acids is probably, you know, certainly much more influenced by gut microflora than other species that don't have such a long gastrointestinal tract and so much opportunity for that microfloral influence. So, you know, there's a reason you don't have DHA enriched beef. It's because as soon as that DHA is going to hit the rumen, the microbes are going to tear it up and you won't have anything left. And I think that in the case of the tilapia, or any fish species or any vertebrate really that has, you know, such extensive gut microfloral
Right.
action, that you may be limited there as well.
Next question from, I think from Korea, from Dr. Ali Hamid Goli.
Yeah,
You
hi, Ollie.
mentioned, you know him, I know him as well,
Yeah, I do.
right? Yeah.
Yeah.
So you mentioned that LC PUFA requirement is related to trophic level of fish. Can we also relate the LC PUFA requirement to the natural ability of fish to retain these fatty acids?
Yeah, so if I understand the question correctly, it's, you know, is the LC PUFA different for a fish recognizing that, you know, that they could really conserve those fatty acids maybe more so
Right,
than other species?
right,
And
right.
yeah, I think so. And I think that's a question that we asked ourselves when we were doing some of that work where, you know, you could reduce the amount of LC PUFA in the diet further without seeing growth suppression when you had a diet that was rich in saturates or monounsaturates. And the way that we described that is we said, well, look, it's an apparent reduction in the absolute requirement. Is it actually changing biochemical demand for LC PUFA? I'm sure not. I'm sure the demand of the animal is the same. It's just that they're able to satisfy that with less inputs because they are using that that selective retention or whatever mechanism you wanted to use to describe that selective accumulation. So yeah, I think Ollie's right that it's when we talk about being able to meet the requirements more efficiently, it's not that we've reduced the requirement per se. We're just giving the animal the best chance at meeting it efficiently and utilizing, harnessing that ability to retain those LC PUFA.
Thank you. And next question is from Dr. Kevin Fitzsimmons. You know Kevin very well, I guess.
Of course, yeah.
As we are with the question, we're going to a different realm, the algae industry, right? So said Corbin and Veramaris claim they have scaled up and compete with similar prices with fish oil. Do you think the algae source fatty acids will continue to gain market share, or do you foresee nutritional impediments?
So I think right now you're dealing with both, right? So you've got the economic limitation, which, you know, I think it's there's one thing that will resolve that, and that is, I guess, rising prices for fish oil. You know, I'm old enough to remember when things like soy protein concentrate were, you would never consider a protein concentrate in feeds. It's far too expensive. Well, then fish meal prices go up and suddenly it's competitive. So, you know, I think that will only help the algae sector because while the price of fish oil fluctuates from year to year, I don't think there's any reason to think that the price is on average going to go down. So as that price continues to climb, then perhaps hopefully the cost of production on the algal front is coming down at the same time. So, you know, hopefully they can kind of meet somewhere in the middle and be more competitive there. But in terms of, so that's the economic limitation, but then there's also this utilization or functional limitation that I think Kevin's alluding to, in that, you know, single-celled algae in particular are really tough little critters, and they're not readily digested, they're not really broken up, and when you're dealing with small unicellular material, how do you break that up further in a way that isn't hugely energetically expensive, hugely costly, because again, that's just going to drive those prices back up. So I'm not an engineer, and I won't even attempt to play one on TV, but I do know there's a lot of smart ones out there. So I think if we, as a nutrition community, can say, here are the problems that we have with these particular ingredients. We need to have the cells fractionated differently, or we need to have this particular element of them physically broken up, or whatever the limitation is that we identify that would get in the way of further utilization. I think there's a lot of really brilliant chemists and engineers out there that would be more than happy to help us.
Excellent. I think related to this, like the lipid source, there's a question from Mr. Chiranjeev Pradhan. Can insect lipids be used in fish feed? I know yes, but what are the positives and negatives? And we can talk a little bit about lauric acid. We hear a lot about lauric acids, right? Medium chain fatty acids. So if you can,
no, as you say, Kabir, the short answer is yes, you can. You know, I think as an energy source, they're great. I'm actually more interested in, again, that kind of lipid class composition from insect lipids, maybe even more so than I am in the specific fatty acid levels, because I wouldn't say that the fatty acid levels the profiles are particularly noteworthy. There's
Right.
certainly a lot of things out there that look kind of like insect lipids, but especially working with species that are naturally insectivorous, like trout, at least for one part of their life stages. I think there's probably a lot of potential benefit for inclusion of not just those lipids, but maybe just those whole meals and things like that as well. Because you know, a rainbow trout, if you look at their gastrointestinal tract, they're kind of built to deal with difficult lipids, and there may be even some kind of, I guess, kind of unconsidered at this point, you know, nutraceutical types effects. You know, that when we look at the natural diet of an animal and then what we put in the feed, a lot of times there's a mismatch. And you know, how many times have we seen that, you know, oh gosh, when we put in the diet what they evolved to consume, they do a lot better, and not just better in terms of growth, but better in terms of, you know, disease resistance and stress tolerance and all these other kind of
Right.
intangibles. Although really they shouldn't be considered intangibles, because things like stress and disease resistance and all of that certainly affects the bottom line. So, so yeah, I think there's a lot of interesting work that's going to come out in PES and will continue to come out related to insect ingredients, particularly on that kind of microbiome and nutraceutical type of research.
Right. Next question is from Dr. Zuri the American. I don't know whether you know, she's the editor of Aquaculture Asia Pacific magazine.
Oh, yeah, of course.
Okay, as organizer of TARS as well. The question is, it's about ARA, and she said you did not say much about ARA. How important is this for any specific species?
Okay, so it's guilty as charged. I didn't talk about N6 pupa as much today. I already talked too much. I was given 30 minutes, and I think I went quite a bit longer than that. But no, I think, yeah, ARA is hugely important. And actually, in some of our work with California yellowtail, we kind of expanded that experimental design where we were looking at just the combinations of EPA and DHA and trying to understand the relationship between those two. When I talked about that, we did another study that had ARA in the mix as well. And in that case, the answer was essentially that EPA still wasn't very important, but DHA certainly was, and so was ARA. And to really maximize the growth performance, you needed to have both. So in the studies that I presented here today, obviously we had ARA in the diets, we just weren't manipulating that one. In this other project, we did. And yeah, it was hugely important in that case. And yeah, for cost or time savings, I didn't talk much about ARA, but it is hugely influential.
Right. So I call them together as DHARA, DHARA, right? So the DHARA in if you go by like Sanskrit, right? All Sanskrit, DHARA means flow. So to have a right flow, you know, it's like DH and ARA. That's kind of
perfect. Perfect. I like it.
So another question from her is, does the requirement of C18 versus LC PUFA change for broodstock diets?
Oh, okay, yeah. So, so we did a little bit of broodstock work back in the day, looking at many of these same concepts. You know, what are the requirements for growth of broodstock? What are the requirements for maximum fecundity? What are the requirements in terms of you know, ultimately larval success? However you define that, whether that's just the absolute numbers of surviving fry or their size at transition points or what have you. So, and I guess maybe instead of referring to that as requirements, really we were just comparing different oil sources, but using different oil sources to try to gauge what the influence of different types of fatty acids were. Trying to, in many cases, to apply the work that had been done by, you know, folks at Sterling with Atlantic salmon and other species, applying that to some of our freshwater fish. And what we saw was actually pretty strikingly similar. The effect of saturates, for example, when you were feeding a diet that had basically a little bit of LC PUFA and a whole lot of saturates, you ended up with much better quality fry, better fecundities, better larval survival, better success overall than you did if you fed the diet that had the same amount of LC PUFA, but you otherwise loaded it up with those C18 PUFA. And I think that's interesting because, you know, it shows that this, whatever this phenomenon is, where overloading the system with C18 PUFA, it's almost like it creates metabolic confusion for the animal. They don't know what to do with it, so they just kind of stick it everywhere, and it finds its way into the tissues and creates problems. It seems to be true through all these different processes, whether we're talking about just synthesis of new tissues for growth, lean growth, or whether we're talking about the synthesis of new tissues like gametes. So we saw quite a few parallels there.
Excellent. The next question, we have a lot of questions today. Sorry,
Whew.
we have time. We have time. Sorry, we have another 10-15
No,
minutes
this
to
is
go.
wonderful. I really appreciate all the interest. This is great.
It's from Dr. Alexander Chong. So he said, very nice diagram to map the influence of trophic level on LC PUFA requirements of different species occupying different ecological niches. How would you view the relative positions of a freshwater predator, right? Pike or snakehead. Snakehead is a very important species in some part of Asia,
Yeah.
and or African catfish as well. If you can look at that, a carnivorous species, highly carnivorous species, and marine herbivores in the diagram. Okay, I will present the diagram.
Yeah.
Okay.
Yeah, so some of those species are included there. So in that diagram, not the one with the cute fish pictures, but the other very complicated kind of ugly one, that includes those species. So it includes some of those freshwater, really carnivorous species, and the marine, more herbivorous species. It's difficult to find information on some of these species. That's the tough thing, is that I really wanted to look at, what are the effects, what are the responses in some of these marine herbivores. And now increasingly some of that literature is starting to come out, which is really exciting. And again, it doesn't all perfectly align with this concept. There's a pretty clear demarcation in my mind between, at least the high and very low trophic level species. And there's some gray area, there's some overlap in the middle. And I think that, there's always going to be the exceptions to the rule, right? But I think our point in putting forward this kind of paradigm of using trophic level is, it may not be perfect, but it's closer than some of these other rules that we've been using, where it's almost a 50-50 whether or not it works, whether or not it's an accurate predictor of what the fish actually need. So I'm sure that someone's going to do some work out there with some strange species to show that it doesn't align to this concept. But I think so far, at least for the species that we have information for, it seems to kind of harmonize things pretty well.
So what are the gaps you think, like in from trophic level? What would be the next step, right, to refine this notion?
Yeah, so I would say that there's probably, well, I mean, if I could do all 30,000 fish species, wouldn't that be wonderful?
Commercially important species, say.
Yeah, but looking at the commercial species,
right.
So one thing that we've never looked at, well, when I say we, our team that was putting together this work, we never really considered crustaceans, because that's pretty far outside of what we normally do. So I would be really interested to see how crustaceans rank in this, because at least based on their biology, they should fall into line with those low trophic level species. But there's obviously carnivorous crustaceans out there as well. So I think that would be really interesting to look at that whole grouping of animals as part of the aquaculture area. But beyond that, I think we talked about how there isn't a lot of work for marine species that aren't those high level carnivores. But beyond things like channel catfish, like tilapia, there aren't a huge number of studies done with those kind of herbivorous or very low level carnivores either. There's just not as much of that work done. At carp certainly, but I think it would be great to fill in some of those gaps as well.
Right.
But
I think this was just a right answer leading to this question, which is about shrimp again, from Dr. Fusigo.
Okay.
Dr. Fusigo, great works in fish. Good to learn that DHA can be converted to EPA by some fishes. Is there similar work done in shrimp? I think you almost answered this question. You can go further a little bit.
I have no idea. I don't read the nutrition literature
in shrimp. Okay, so I will try to find Dr. Fusi. Thank you, Albert.
Yeah.
For joining in.
Yeah, maybe somebody else has maybe someone else knows something about, they could put it into the chat or something like that if they're familiar with the crustacean literature. But I have my hands full with finfish, so I haven't kept up with that work as much.
So to the audience, if you have any idea on this, Albert, please go ahead. Sorry, Albert, we missed you.
You're on mute still.
Ah, okay.
Go ahead.
There we go.
Am I unmuted? Okay, thank you.
Yes.
I come a bit from the old school, but in the mid 80s, we produced a PhD student, Kok Leong Wee, at Sterling University, that I helped to supervise. We published a paper with Colin Cowie where we showed that in snakeheads, Channa striata, it had this very limited ability to produce ARA, which fits in with its position at the end of the food chain. The important point, I think, is just that, you know, whatever species we grow, they need nutrients, not ingredients. And so things like fish oil, yes, it has EPA, yes, it has DHA, but it also has phospholipids, it also has cholesterol, it also has other nutrients. So in species like cobia, you know, we've shown more or less now that probably for highly carnivorous species, probably cholesterol is essential for these species as well. So it's important that we bear all this in mind. Just rambling a bit, but my interest always is from the point of view of the fish or the crustacean in terms of the pathology. You know, I mean, I first came across this in the late 80s in Indonesia, where we had a project growing barramundi, larvae, fingerlings. And there you had a problem where if you hit the side of the tank in a hard way, you would get a shock syndrome and they all die. And at the end of the day, that was just due to the source of Artemia that the project was using, and the fact that Artemia has no DHA. So that's why you have to enrich it. But at the end of the day, but the shock syndrome is something that's very common in many of the species that we grow. Another time where I came across the hufa and their importance was, you know, I was having a postdoc again in the late 80s in Stirling, and we had a person that just finished doing a PhD on herring larvae. And what he showed is that with a deficiency of DHA, their schooling behavior is completely destroyed.
So it's like
Exactly the same thing happens with yellowtail.
the endocrine disruption.
Yeah, and the interesting thing is that one of the richest sources of DHA is the eye. And that's one of the first parts of the body that other predators go on. You know, that even my wife that's consumed so much fish, the first thing she goes for is the eye.
Me too.
So it's a really important phenomenon. But the important thing is that, you know, I loved your presentation. It was really, really important and very, very clearly done. But the other side of the story is what happens when we don't have the right PUFA or arachidonic acid levels. And what we've shown is that it really affects the immune system of the species. And also in terms of oxidized oil, it also can cause leaky guts, which is also caused by saponins, which is also caused by mycotoxins, you know, and so all these things are related. And you're the same as me. I spend my life formulating feeds for companies, and we have to, you know, but we have to think of all the nutrients together. Yes, we know that the fatty acids are important, and the price of lipid is much more expensive than protein at the moment.
Right. So that's where it comes like. Sorry, go ahead.
I was just going to comment on something that Albert said there that I think is really, really critical. And forget about all the stuff that I said, pay attention to this one thing that Albert said, and that is that, you know, it's paying attention to those little nuanced things that are happening with your fish. Like, we can do really sophisticated science, and, you know, already just in my relatively short career, some of the things that are possible analytically are way past anything that I was doing when I was learning as a student, and, you know, I can barely keep up with the technology, but nothing ever takes the place of a fish culturist who's paying attention. And the fish are trying to tell you when something's wrong. It's just, are you listening? are you trying to understand the language that they're
Right.
speaking? And so I think that that's so important. So especially for the students that are maybe listening to this presentation now or later on, like, it's really important. Do all that cool analytical stuff, learn all these tools, but don't forget those fundamentals. Spend the extra hour watching your fish when you're feeding. Like, really watch them, look for those responses, look for that startle response, that shock response, or whatever it is that they're trying to show you.
Albert?
No, ditto, I agree. I mean, my first lecture normally to the students, or when I get the opportunity to give lectures, is to say that whatever it is we're growing for millennia, it's used to a certain type of diet.
Right.
And so I think what's one of the really important things to do that we always tend to ignore is looking at the natural feeding habits of the species in question, which is basically the trophic level.
Exactly.
And the adaptation to that certain diet. A good example is cobia. In Spanish, it's called the pez cangrejo. It feeds on crustaceans. In the wild, it feeds on the bottom. So again, that can also give us a lot of clues into the type of nutrients that it might require. But that's why, you know, praise you that you brought in the trophic level, because it really is all about the adaptation of an animal to a certain type of diet.
Right. We like to think that we're very smart, and to a certain extent we are, but not compared to Mother Nature. So.
Oh yeah.
So this brings like stress or shock in animals and how to deal with the stress, and we are in fatty acid realm. It brings to the discussion on short chain fatty acid we never talked today. There are a lot of work being done on like 3 short chain fatty acids like butyric acid, propionic acid, acetic acid, and their effect in I think in managing stress of the animals. And there are a lot of there are free acids or encapsulated acids so that you can have a lower concentration or not affecting the gut intestinal pH in the proximal intestine. So any idea or any comments on that? Right, we are still in the fatty acid.
Well, yeah, I'm interested in everything, but whether I'm knowledgeable about it or not is a different story. But I think a lot of this work is kind of like much of the research that's happening right now is increasingly focused on the microbiome, because of course we're not just feeding the organism that's our target organism, we're also feeding all of the critters that are occupying its gut. And so I think there's, obviously a relationship there between, what are you feeding the animal versus what is the animal actually producing. I think one of the things that is maybe a limitation with some of these research projects is a really strong emphasis, obviously, on comparing, the microbiome on this particular diet or that particular diet. And that's obviously relevant, but how do you know which one's right? What is a good control in those cases? Should you be looking at wild fish that have never seen a commercially manufactured feed in their entire life to kind of look at what a real normal might look like for the microbiome for the short chain fatty acid production and so forth? And I don't have an answer to that. Again, I'm not a subject matter expert in that arena, but it does strike me that that's relevant to consider. That if we're only looking at the response of fish to these diets, we may be missing something.
Albert?
Yes. Yeah, I mean, just to add on to what Jesse's, in your presentation, you talked about the interesting results of saturated fatty acids compared with unsaturated fatty acids, and the fact that when we talk about physiological function, we should also remember that the fish also, or the crustacean, can also use them as an energy source. As a much richer energy source than other nutrients. Certainly short chain fatty acids, medium chain fatty acids. In terms of ruminant nutrition, yeah, a lot of work done on volatile fatty acids, and they're important to the health of the rumen. And for sure, they're going to be important within aquaculture species. But also what's interesting is that some medium chain fatty acids have other functional properties apart from serving as a nutrient, but also in terms of parasites, against parasites or against, everybody is looking for something that can stop sea lice growing on the outside of the salmon. And so these things are important. So again, a lot of work needs to be done. But like Jesse will know, at the end of the day, it all depends on the experimental design and how you set up the experiment, what it is you want to show. Yeah, that's my comment.
We have 1 minute left. In response to our call for can discover studies in crustaceans, Dr. Alexander Chong responded that only 2 papers so far demonstrated functional elongase in crustaceans published this year, and both are in crabs, one in mud crab and other was in marine crab.
Okay, well, so we're not too far behind. Those papers just came out this year.
So our audience is there. They're responding very well. Thank you so much. Thank you, Dr.
Great,
Chong.
thank you.
Yeah, I think we are almost at the end of our session. And I think I should thank Jeffo as well again to help us to organize this session. And Dr. Trushenski to be here with a nice presentation and then thoughtful presentations. Albert and I would like to thank you so much. And Albert as well, always an inspiration to do things that matters. Okay. Sorry, it's don't take it otherwise. This is from her. Our next session will be December 10. I think we will bring someone from feed industry as well, from Asia, and we'll talk more about developing feed and raw material ingredient issues with the feed industry. So thank you all again. Hope to see you in the next one. Albert, any last word?
No, just again how much I enjoyed Jesse's presentation. Again, talking about a difficult subject, but in a way that's really easy to understand. If I could just add a little thing right at the end, and that is that I have a new publication, a book coming out soon, but it's on nutritional pathology, where I talk about basically all the different symptoms and deficiency signs or toxicity signs or effects of oxidized oil on different fish and crustacean species, where I try to put everything together, but just really illustrating the importance of these nutrients in the diet.
Thank you so much.
Where is it going to be? Yeah. When and where can we find it?
It's going to be published apparently by 5M.
Huh.
5 months, right?
Excellent.
Yeah, they have a series. Right. Anyway, they have a series in Europe on pathology, and so apparently they want to publish it. So
wonderful.
Let's see.
We will share a publishing house then. Okay. And if I may also just thank you both and everyone for calling in and being a part of this, and for your questions, and for really giving me the distinct honor of being a part of this web series. And great job moderating, Kabir. Thank you. That was a lot of questions to field, so I appreciate it.
No, thank you. I covered all the questions we had. I think.
It was a lot. We better stop before there's more.
I should say thank you to Pumjai in the back, right, helping us.
Of course.
Yes.
Thank you.
No, thank you to everybody.
Okay. Yeah, thank you. Thank you all. Take care.
Bye-bye.
Bye, everyone.
Bye,
yes,
take care.
See you next time. Bye-bye.